Yeah, good afternoon. So you heard Liz Ainsburg. She's very sorry to be not here. So and she doesn't feel well, and also her daughter is sick, and she sends you all the very best wishes, and she tries to get to be involved in the discussion on Friday and remotely, so hopefully it will work. And yeah, so I will present something about the LDLensRad project. So I will introduce you to the motivation behind this project and the aims of this project, as well as the research methodology. So will it work like this? No. Okay. So yeah. Yeah. What? Yeah. So yeah, it's about cataracts. So, but we are not dealing with humans. Even if these examples are human eyes. So, you know, cataracts are the most frequent cause of blindness worldwide. And yeah, cataract, it's caused by a multifactorial etiology. So, main causes are age, genetics, so maybe congenital cataracts, as it's seen in this boy. Okay. Should I go closer to the microphone? And for radiation cataracts, as you heard before, so the main type of cataract is the posterior subcapsular cataract, which can be seen here, and it's a nice picture here from Norman Kleiman. So here you can see this clouding of the lens in the really back part, so in the posterior part. So, but what really causes cataracts? So it's incorrect amalgamation of the lens fiber cells and wrong deposit of proteins in the lens. And yeah, that's, oops. Okay, that's what we want to look at. And so the different causes for cataracts, as I already said, and one for sure is ionizing radiation. And the lens seems to be much more sensible than thought before. So ICRP reduced the threshold for radiation to 0.5 gray for the lens. And so occupational lens dose limit now is at 20 millisieverts per year. And over 5 years, no single dose per year should be exceeded over 50 millisievert. But all these data is mainly based on epidemiological data. So the threshold is set because of epidemiological data point to this threshold. And so ICRP realizes that mechanistical data are nearly missing. And that's what we want to do with the mice. So, how could we start? So what do we know? So we have a good understanding of lens development, of the biology of the lens, so the lens structure, the physiology of the lens, and so the procedure, how lens fiber cells are formed. And so we for sure know from the epidemiological studies that ionizing radiation can cause cataracts, especially the posterior subcapsular cataracts. And mainly from high doses. So with the lower doses, it's still in question where the threshold really is. So that's what you heard before. And how big is the involvement of the genomic damage? So we know there is an involvement of genetic background, and the genomic damage in the lens, so especially of the epithelial cells. So how big is this involvement? So how does the morphological changes look like? And we know from mouse experiments so that the genetic background is one important part, or could be an important part. So of the sensitivity to cataracts. So cataracts are for sure age dependent. And another thing is the detection of cataracts. So there are different methods. I will come to this later. And one thing is, so what don't we know? So, as I said, the mechanisms are not very well known, how cataracts develop. So we know some of the reasons. So we know that diabetes, age, gender could be a reason higher for a higher risk to develop cataracts, and but also diet, and so on, smoking, drinking. There are different reasons, but we don't know how the real mechanism is, how cataracts are built. And we also do not know the real mechanisms for lower doses of irradiation. So maybe there are different to higher doses. And we want to look for research models, and research models in human and animals. So human cases were already presented, and also in cell culture. So, I think, here this was what was nearly, already presented before. So from the clinics, we have some cases where to look at, and so we want to look at the different types of cataracts. So the nuclear cortical cataracts and posterior subcapsular, and what could be important is the latency period, and this could be addressed by mouse models. We want to do modeling with the results, and Liz put here some of the confounding factors. I don't know what she want to really say with this. So there are a lot of confounding factors for the development of cataracts. So I already mentioned a lot of them. And our main factor is the ionizing radiation. So that's our study, so the LDLensRad study, so towards a full mechanistic understanding of low-dose radiation-induced cataracts. And so we want to look for low-dose cataracts in the mouse models. And this is a project of the first concert call and to the first question. So we want to address how irradiation promotes the development of the cataracts. And so mainly what is the influence of dose and dose rate, so also in low doses, and what's the influence of genetic background, the age and the sex. And so with the results we want to address the question if lens opacity can really be a biomarker for radiosensitivity. That's the project outline. We have a lot of in vivo experiments, so with our mouse models. So for the mouse models, we use different mouse strains, so mice with different strain backgrounds, so BLIC6 mice, 129 CD1 mice, and so on. And we also have 2 mutant mice, so heterozygous ERCC2 mice and the patched mice. So to look for the genetic background differences, and with these in vivo experiments we want to address short-term investigations and lifetime studies. So these mice are irradiated with different doses, so under a cobalt source. So we have for the control group 0.5 group, 1 gray and 2 gray group. And these mice will be examined over a period of 18 months after irradiation. So irradiation time point is 10 weeks of age of the mice, so that means young adults in the case of mice. And we're doing these in vivo examinations, so monthly Scheimpflug examination to look for the lens density over the time and the cataract development in these lifetime studies. So we want to look at the lens physiology, also doing after 18 months when the experiments are finished. So organ pathology, so if the mice died before, or how many cancers build up, or different organ pathologies occurred. And we also check for behavioral effects, so cognitive effects in these mice. And there are some short-term groups, so some of these mice are examined earlier time points, so that means 6 and 12 months after irradiation. And in these mice, we look for DNA damage, so proliferation, biochemical effects, so the morphology of the eyes, oxidative stress will be addressed. So the intracellular communication between the lens cells. We want to model the genetic pathway analysis and look for microRNAs. So, and next generation sequencing. So, and there's also parts, so for in vitro experiments in the cell culture, so different primary human lens epithelial cells are examined, and also different cell cultures from mice, from different strain background, and with different mutations. There are similar endpoints for the DNA damage, proliferation, morphology, and we will look for oxidative stress and so on. And so one important point is we want to look for different dose rates also in the mice, and those rates are 0.063 gray per minute, and the higher dose rate is 0.3 gray per minute. And we're doing this for the mice and also in the cell culture. And for cell culture, the doses are really going lower to 0.05 gray. And important is we want to do a statistic modeling, and with the results, and bring together all these and answer the outlined questions. So for the project, we also have an advisory board who helps us to address the right questions and with the experimental design. So these are first initial results. So very initial results. So this is where we look for key influences like age and genetic background. So this is Scheimpflug examinations of the mice with the mean lens density here in percent and time after irradiation. So these were mice irradiated at the age of 10 weeks. So in a wild type background, but hybrid mice, F1 mice, Black 6 and C3H mice. And as you see, so the curves go up, so the lens density increases over time. But as you see, in all different groups. So this is a 0.5 gray group, the red ones, and 1 gray group, the green, and the violet dots, it's 2 gray group. So you see these groups are really close together, and so that means that's not a big difference between irradiated and unirradiated cohorts, and also the unirradiated mice. So at least statistically, there's a significance, but on the clinical side, so we have here maximum density and the mean density of 7 to 7.5%. So that's not really clinically relevant. And but as you can see here in this picture, so the full signs here are from patch 1 mutant heterozygous mutant mice. So, and you can see the mean lens density of these heterozygous mice is higher than from the wild types. So from Black 6, it's the lowest. These CD1 wild types. This is F1 mice from a mixed background, and these are the heterozygous patched mice. So you really see in this mutant the lens density goes up. So these are some examples from the mechanistic data. So from the DNA damage dose rate effect for lens epithelial cells here in the upper row. So these lens epithelial cells were divided, were stained by immunostaining with the 53 BP1 marker, so a marker for the double-strand breaks. And you see in the central region of the lens the number of double-strand breaks, so the foci number goes up with the dose. So we have the dose of 0.5, 1, and 2 gray here. And we have these two different dose rates of 0.3 gray here, the blue line, and the 0.063 gray per minute, the red line. And also for the lens epithelial cells in the peripheral region of the lens. So it looks somehow similar, and what's really obvious that the lower dose rate gives more double strand breaks in here. That's also true for peripheral lymphocytes, which you can see here. So the darker bars represent the 0.3 gray per minute, whereas the lighter bars represent the 0.063 gray per minute, and this is the control. So in both irradiated groups, the number of double-strand breaks is much higher, but it's even higher with the here it's here it's a different way, so it's a bit higher in the with a higher dose rate. For the patched 1 mice, we have the foci number in here, so yes, that's the same like in the black 6 mice we've seen on the top here. So the dark gray is 0.3 gray per minute, and this is 0.063 gray per minute. So with the lower dose rate, we have more foci. So the ionizing radiation also influences the hippocampal region, neurogenesis, and the behavior of the mice. These are some data from the patched mice. So this is done at INEA Institute. So the patched mice on the have the differences from patched mice on the CD1 background and black 6 background, and with different dose rates here, 0.3 and the 0.0363, so the lower dose rate. It's again much higher, and on the black 6, so the CD1 CD1 background gives even higher results here. So it's a difference between the background. So maybe that's important. And also Y-maze entries, so that's a behavioral test from the Y-maze, so that's a Y where you look for the entries of the different arms of the mice. And these are the control group mice, and the mutant mice have much more entries, and at the 2 gray group it's highly significant, but also in the 0.5 gray group. And we have this INSTRA project. I will tell you something about this later. So it's a previous experiment where we started with these mice experiments, and also behavioral experiments were done. So a lot more data collection and analysis will be done on the data we receive in this LDLensRad project. So with the microRNA and pathway analysis, so lipidomics will be done, and mitigators of exposure. A lot of eye lens histology will be done when the long-term experiments with the mice finish. And I will show you some more detailed data of these OCT examinations later. And so also a lot of in vitro studies will be done to support the in vivo data. So yeah, let's come to the conclusion for this, for our project outline. And it is anticipated that the LDLensRad will have some key implications for radiation research and protection. And so the outcomes we're hoping to define is the shape of the dose response. So that's not really clear for low dose irradiations at the moment. So we'll clarify dose rate effects and the influence of genetics, age, sex, and some other factors. And so hopefully advancing the knowledge about the risk of radiation cataracts at low doses. And with that, there was a stakeholder meeting, and there were also clinicians present, and they already overtook something to their work, so they now know it's a risk, and so they started to wear these eye shielding glasses. And so up to now, we had already had a lot of presentations of this LDLensRad project in more than 20 international meetings. And yeah, I want to thank all the partners busy in the LDLensRad project and the INSRA project. So the partners from the INSRA project are marked with a star in here. And yeah, many thanks for Liz Ainsburg. She's our project leader. And yeah, she gave me her slides, so maybe she would have done better, I don't know. I think so, but I received these slides yesterday in the afternoon, and but I hopefully could have clarified something. Oops, maybe this is Liz's email address. So should I go on immediately with the next presentation? It's okay.
Well, thank you very much, Claudia. Maybe we have time to take, not sure we will take 3 questions. May I suggest also to you the email address of Liz if you have a burning question for her, since she will be able to participate in the discussion remotely on Friday.
Well, my question concerns the choice of the mice. You select 2 types of mutated mice, the patch mice and the ERCC2 mice. Why do you select the mutation of those genes? Do you have some hypothesis in the mechanism underlying the radiosensitivity eyes?
For the patched heterozygous mice, it was already known that they are more sensible to cataracts development, and we thought this could be a good model to get results for the mechanism of cataract development. And so the ERCC2 gene is involved in DNA repair. So we thought this could have an impact. So obviously from this Scheimpflug data, it is not that involved, but we didn't know before we started.
Okay, thank you, Claudia. We have a question from Susanna and then Mark for a short question.
I have a question concerning the radiation. Do you irradiate only the eye of the mice, or it's a whole body exposure?
It's a total body exposure, so whole body is irradiated. So we all use a cobalt source in all 3 locations where mice are irradiated, and we use the same doses and dose rates. Yeah.
And concerning the DNA damage dose rate, you show after 4 hours. Do you know if the double strand break maintains later or it's natural repaired? After 24 hours, do you have a natural repair or do you have an accumulation of damage with certain doses?
We do not have the data at the moment, so this will come. And this is experiments from lens epithelial explants, so not really in the whole mice to look for these DNA damage. But yeah, at the moment we just have data for these 4 hours.
Okay, thank you, Claudia. Mark, for a final question, short question.
Well, following on from that, I was just wondering, what is the relevance of DNA damage to cataracts? Is it known what's you're measuring it at 4 hours, and the cataract is then develops 18 months later? I mean, DNA damage we know is related to radiation exposure. I mean, but you could measure that in blood. So and you're measuring it in the eye, but so what? It's just a marker of exposure.
Yeah, it's not really known. That's why we're doing these experiments. But there's a hypothesis that because in epithelial cells you have still the cell nucleus and the DNA. And if you have some DNA damage there, so that might lead to a wrong development of lens fiber cells. So or maybe so, especially for posterior subcapsular cataracts, there's a hypothesis that cell nuclei are not degraded. And move to the back of the lens. So, and if you still have cells with the cell nuclei, so they are not clear. So then, yeah, you have cataract.
About this DNA issue. So, in my talk, I mentioned briefly that there is a collection of micronuclei samples which are scanned. Recorded and are available for at least visual inspection and analysis. Very aware of that, and did you try to access this data set?
No, we were not aware of this, and yeah, we're doing it.
Keep in mind.
Yeah, thank you very much.